with that, we're going to start our presentation so that NASA can absorb a little bit of information from all of our speakers. And the first speaker is going to be Julie Segre from the National Institutes of Health. Julie, please.
Okay. Thanks, Mark. Okay. I, like you, wasn't really sure what I was supposed to talk about, so I just picked 3 stories and can go from here in greater depth. Okay. I'm going to tell you 3 stories. I'm not going to even bother, going through all the definitions of what is microbiome, If you didn't, I hope that was discussed on your bus rides over. So I'm going to talk to you about tracking Candida auris, which is a new emerging fungal pathogen. I'm going to talk to you our story about the hospital infrastructure with Sphingomonas carinensis. And in terms of the human skin microbiome, I'm going to talk about our work on eukaryotic viruses. Okay. Take you back in time 10 years. We did the first survey of body sites of the skin, using this as an ecologic niche to understand that the microbiome is dependent on the body site. So we are all this is the topographic distribution, which basically is that, the bend of my left elbow is most similar to the bend of my right. And after that, the bend of my left elbow is more similar to Jack's than it would be to my underarm, which is like a sweaty environment with large hair follicles. So the microbes is really all about what is the nutrients that are available for the bacteria, for the fungi, what are all the nutrients that are available to them? And the skin is rather a desert where there isn't much to eat. It's not like the gut. So these are very limited nutrients. I think that does give us a lot of opportunities to think about prebiotic solutions. We also did an analysis of the fungi on the skin, this was with amplicon sequencing with ITS1, and found that the body is these are younger healthy volunteers, so 20s, people in their 20s, 30s, Malassezia dominated. And the only place we saw fungal diversity was on the feet, which wouldn't really surprise you considering that what we really see is toenail infections and athlete's foot. That's where you're seeing the fungal diversity. Okay, I'm gonna move really quickly. So we've been working for the last 5 years, probably, well, 3 years on Candida auris, which is an emerging fungal pathogen. You don't get many of these. I mean, so what's been interesting to us is that Candida auris was first detected only 10 years ago by the CDC and there were a few isolates that were misidentified as Candida haemonia, Candida lusitaneus, things that don't typically cause bloodstream infections, and that caused the CDC to begin to investigate. And we found that these were a newly identified species. And the first one was found in Japan in the ear canal of a patient, but then we pretty quickly identified Candida auris on 4 different continents: South Asia, East Asia, Africa, and South America. We also got engaged because the third patient in the US was identified at the NIH Clinical Center, which really jump-started all of our issues. I'm going to talk to you today about the outbreaks that we've been studying in Chicago and New York. We talk a lot about antibiotic-resistant organisms, but I thought this would be a particularly interesting case because you would not screen for this, right? But here it is, and 10 years later, we now consider this an emerging pathogen. When we put together the CARB initiative for combating antibiotic-resistant bacteria, the one thing that was of concern are these azole-resistant Candida, but I would say that Candida auris, that was sort of Candida albicans. Candida auris is just like moving this whole field really quickly because Candida auris has extremely high levels of antimicrobial resistance, causes difficult to treat bloodstream infections. Our work has really been that patients remain colonized on their skin for months to years. We have no way to decolonize these patients. They shed Candida auris into the environment where it persists and can be, as viable. Originally, the testing required 7 days of culturing on selective media. We've now moved that to like a PCR and a sequencing assay, but I would still say culturing is the gold standard because those other methods can't really distinguish live from dead microbes. Okay, so this is, I think this is fascinating. So you have it evolving. So you have Candida auris emerging on these 3 different places. And these are highly clonal. So if I sequence isolates from the South Asian clade, genome sequencing, they're within you can see here, they're within like 10 to 100 SNPs of each other. But the 4 different isolates are all like thousands of SNPs different from each other. So it really does seem like it evolved independently 4 times. But we don't understand where did it come from. And there's all these ideas about maybe this is because of azole resistance and the increased use of azoles as it came off patent or being used prophylactically for different at-risk patient populations. But none of these theories make sense. We have done extensive searches of SRA with Kamer-based approaches, and we can't find Candida auris anywhere having been deposited. We've also gone back to all of our patient samples, people who came into the NIH Clinical Center, and we can't find it in any of those before 2017. So if you find it, let me know. This is just quickly, this is an example, of how this could happen, and I'm trying to give this as an example if you would ever think about, what could possibly go horribly wrong. So patient 1, patient 2, this is a classic story. We've had this at the clinical center, but in terms of this illustrates it very nicely in Chicago. They had patient 1, patient 2. There was a link between those 2 patients. Then they did surveillance for months and got, nothing. And we did the sequencing of those patients. So this is 10 patients from Illinois. This is one of them where Candida auris is this red. The New York was already much further. These bar charts, I'm showing you relative abundance 0 to 100 of the ITS1 region. And up top from Susan Wong are samples from skilled nursing facility patients in California where there's at that time, that is no longer true, there was no Candida auris there. And you can see the taupe color now is Malassezia. That should be your dominant skin microbe. You can see already in these patients, which we found very unexpected, these are inguinal, so groin. It can convert over to being Candida, which I didn't think of that as, it wouldn't be totally surprising, but I wouldn't have thought of that as a skin commensal. But in the Illinois, we could get the first sequence of the patient, and New York was already much further along. These patients were heavily colonized, and so as an example, in Illinois, 2017, they've got those 2 positive patients, and then they do 2 negative point prevalence surveys, and we all think it's good. But then 2018, quarter 1, quarter 2, it starts to tick up, and you've got new clinical cases, surveillance cases. And by now, we're kind of holding steady, where there's 10 to 20 new clinical cases and 20 to 40 new patients picked up on surveillance. Of great concern is that they're azole resistant, amphotericin B, and echinocardin. And we really have seen the triple resistant already here in the US. So we're even just trying to figure out, this is how you do a study from the very beginning. What is the site of colonization? So that's a really important factor. When we had a carbapenem-resistant Klebpneumo outbreak at the NIH Clinical Center, at first we were doing inguinal crease axilla groin surveillance and missed the first transmissions. So we've been trying to figure out what is the site of colonization, and axilla groin was the site that the CDC is doing surveillance on. We also found a fairly high burden on colonization in the nares. And we also found it, these are nursing home patients. We found it on their hands and on their feet, which probably has to do with the environmental. But we actually, when you look at that, we actually couldn't find, like when you search for, Staph aureus or, you can swab inside the nose and pretty much get most people, and there may be some that you would miss, but a very small percentage. We can't find what's the minimal body site. Example, when we, Illinois and New York, we all think that the nares is an important site to screen, but then California incorporated that into their screening, and they didn't pick up any extra patients, and then we figured out, oh, they have the African strain and we have the South American strain, so there may still be differences due to what strain you have colonizing your patients. This is the kind of sequencing data that we're getting from these patients. So what I'm saying is that, there'll be patients, and like in this one, it's only in the toe web. Now I've switched, it's like this brick red. I'm only gonna find it in the toe web of that patient. This one, the toe web is completely, the toe webs can be completely Candida auris. And, other sites can be mildly colonized. And so we're really digging into this sort of set to see now, really our main question here is, how does Candida auris just become a skin commensal? So we're trying to attack that by looking in mouse models. And so this would be the first experiment, because the question I get from people is like, oh, but does Candida auris really colonize the skin? Or is it just, fecal matter that's colonizing the skin? And I want, in the same way that you think doing science is hard on the ISS, doing clinical trials in nursing homes is also really hard. And there's certain things that just mice are really good for. So in this case, working with Yasmin Belkade, we're doing a study in which we are colonizing wild-type Black 6 competent, just totally wild-type mice on their skin with Candida auris, and as comparison with Candida albicans, about which we know a lot more. We colonize them topically, so it would be like as if you were exposed by, picking it up from the environment. We're going to do superficial swabs, and then we're going to also just look at the entire tissue and see. So Candida albicans at day 14, exactly what you would expect. It's a gut colonizer, so you're going to get it in the small intestine and the colon. Candida auris is a skin colonizer. I mean, that's really where we're going to see it. And, you can pick it up, but it pretty much does go away. I mean, if you're talking about a germ-free mouse or something like that, of course it's going to colonize everything. It's not like it can't colonize the gut, but there is colonization resistance in those areas. And then, the question has been that it seems to persist on the patients, and we're seeing the same thing. So this is what you would be testing. You would be doing a swab, right? And you'd be swabbing the person's skin. And on mice, it seems that by day 14, it's starting to go away. And by day 21, they all would be negative. So you'd think that this mouse had cleared. But when you then look at the tissue, they're staying positive for 2 months. Probably even, we still find positives at 4 months. And this is just like a wild type mouse. So I think that's getting more into, these are the kinds of things that wouldn't exactly surprise you. We see the same thing with, carbapenem-resistant Klebpneumo, that, the patient can go negative, and then you give them antibiotics, and boom, it's back, and it's the same strain. So I think this is something probably in this, what we're trying to talk about here is that there are reservoirs for these kinds of pathogens. So I think I sort of talked about, all of the research that we're doing now, like trying to identify the primary site of colonization, look at the strain specificity. Some things, that's why I was asking our astronaut yesterday, we're really trying to look at the skin bathing protocols and to find out if the chlorhexidine that's being used in these skilled nursing facilities is preventing or driving colonization. Our results in mice preliminarily, I would say, is actually that the chlorhexidine is protecting the mice from colonization. We're trying to just like work with CDC to get all this information out. We do not know where Candida auris came from. We don't know how it persists. But this is like a kind of story of how we're trying to respond and develop really like an integrated systems biology approach to an emerging pathogen, working with our colleagues at CDC and in the skilled nursing facilities in the New York and Illinois State Health Department to sort of look at what's happening in human health and can we build models to understand that. So coming back to the skin microbiome, I wanted to give one story, and I'm gonna have to move probably more quickly. So this is about shotgun. You know, we've talked about amplicon sequencing, which gives you a good idea of who's there, but really to move into shotgun metagenomic sequencing, where we're doing, you know, read assignment and we get the same kind of map, but we're looking at the bacteria, the fungi, and all the kingdoms. What this has really pointed out to us is the inter-individual variation, but the stability. So I'm showing you here 3 different healthy volunteers in the shotgun metagenomics. And you can see that like this person we thought was in the middle of a phage bloom. This is the alar crease, the side of the nose. Because this purple here is a Propionibacterium phage, and we thought like that was totally weird. But you come back like this is time point 2 is a year later and time point 3 is a month after that. That person still like totally looks healthy and normal, but their skin microbial community is dominated by Propionibacterium phage. This would be more typical of what we see, where it's a balance of Propionibacterium and its phage. Some people just are all Propionibacterium with very little phage. This healthy volunteer is our volunteer with the greatest amount of taupe-colored Malassezia. Everyone there's a lot of individuality, but there is a lot of stability to that individuality. And that individuality extends to strains in terms of thinking about, you know, could you identify someone, right? And could, you know, are there strains? Are you going to be able to get a new strain into the community? Because I think a lot of people probably think that like you take a shower, you know, you wake up in the morning, take a shower, and then you're like completely recolonized. No, the microbes are living much deeper into the skin. We probably see about 10,000 bacteria on the surface of the skin if I swab, but when I do a punch biopsy, there's 1 million bacteria there, and they live into the hair follicles and the glands. But if I've color-coded them based on their based on the phylogenetic tree, you can see that the strains of P. acnes would be very distinct for healthy volunteer 2 and 3, and that these are the same 3 time points. So a year later, that person has more or less the same strains and quite distinct from volunteer 3, but they keep those strains. The thing we get out of that I thought might be interesting here, the thing we get out of the shotgun metagenomic data is also what are the eukaryotic viruses. And this is the first look we've had at the eukaryotic viruses because they are in healthy volunteers over there on the right. The eukaryotic viruses would be that purple, and these are actually normalized to genome size. So that's actually very few reads. It's very hard to characterize what they are. But when we look in primary immune deficient patients, a lot of whom are seen at the NIH Clinical Center, which is sort of a center for that, these are 26 patients who have the same, have mutations in the same gene, the dedicator of cytokinesis 8, which is important for the actin cytoskeleton and the immune cells as they migrate through the skin. It's a very collagen-rich environment, and you have to really squeeze through, and these cells kind of shatter as they try to migrate through the skin. So they have very much of a peripheral immune deficiency in terms of the resident immune cells in the skin. And what we're seeing is that the immune system is a shield against eukaryotic viruses. I mean, I would say that the bacterial and the fungal communities on these patients is slightly different, but it's really still the same phylum. Now what we're seeing is an enormous expansion of the eukaryotic viruses that can colonize skin in the absence of resident immune cells. It's a lot of papillomaviruses, molluscum, polyomaviridae, and we can, from these patients' skin microbiome, from the shotgun reads, we actually take the viral reads and the unassigned reads and do a de novo assembly. And from that this is an example of the papillomaviruses, and we can identify hundreds of new HPV types. We also found a new HPV species, which had 25 types. So I think we haven't really explored the eukaryotic virus component of the human microbiome. And as well in this paper, which actually has now been published, we look at the loss and gain of HPVs and how they must be present in the, they must be present somewhere in the environment or on the patients, because we can see them being gained or lost on multiple body sites over the course of months. We also saw RNA viruses, predominantly here in the NAIRS. And I guess this study, in terms of thinking about this, obviously these patients have a profound immune deficiency. We're now doing these kinds of experiments on looking at the skin microbiome of patients going through transplant who are more transiently immune compromised. But we also think about it in terms of the elderly population, in terms of viral, you call it viral reactivation, but I think there's a component of this which is, weakened immunity and the viruses coming out, that we may all harbor many more eukaryotic viruses than we can detect at this point. And then I wanted to wrap up by just talking about something that was an investigation we did at our own NIH clinical center, which I can imagine for you guys on the ISS would be a worst case scenario. I can tell you that even at our clinical center in the ICU transplant wards, this was also a worst case scenario. Okay, so my hospital epidemiologist and I, we meet on a monthly basis and we kind of just review what's happening. This was one of those discussions in which she said, I have 6 patients with clinical cultures of Sphingomonas. I'm sure it's polyclonal, but would you sequence them anyway? Sure. So this was over a year. We probably are always going to have 1 or 2 Sphingomonas at any point during the year. But we sequenced these 6, and the text message I sent back to Tara was, yeah, it's polyclonal, but with, so this is, in terms of what we do, it's sequencing them, assembling the genomes, and then just doing average nucleotide identity. I mean, I think actually this is something, Charles will tell you more about this, but this is something that you guys could really do if you ever needed to. So polyclonal outbreak, but within the cluster, there are 4 of these isolates. So these are the 6 different patients, and 4 of these isolates are, clonal, like over 99.92% average nucleotide identity. So then we started to look like what is the source? What connects these 4 patients, and, running the sort of epi records of, did they have the same, diet? Did they have the same rooms? Did they have this? Did they have that? Sorry, I'm getting ahead of myself. This is just, how we calculated the number of SNPs. I actually thought this was all pretty straightforward to do. We had a PacBio genome. You'd have an Oxford nanopore genome, and then we're just calling SNPs with SNPy, written by Torsten Seaman, looking at Illumina data here. So highly clonal. Sphingomonas, gram-negative, survives in nutrient-poor conditions, found in water in low concentration, typically considered low pathogenicity. But one of the things is when we sequence the genome, I'm sorry, I forgot to mention, the reason that we cared about these isolates is these are highly resistant organisms. So we didn't have, besides sulfatrimethoprim, Bacterium, we didn't have any drugs that these isolates were susceptible to. And we couldn't find even any antibiotic-resistant genes on their, in their genomes. We postulate that these are a class of bacteria which basically, living in nutrient-poor conditions, they basically just pump everything out, and then they'll just selectively import the few nutrients that might be useful. Because if you're living in that hostile of a sort of environment where it's a biofilm and basically everything that goes past you just is, meant to kill you. But that does increase its antibiotic resistance. Okay, so then we, at the clinical center, in terms of these investigations, we are incredibly fortunate to work at this clinical center, which is like a gold mine. I mean, so we went back and did a retrospective analysis of all Sphingomonas patient isolates. And we store all these isolates. So we then retested them all by MALDI and sequenced them and found that our first isolate that was Sphingomonas carinensis was in 2006, which was right after the hospital opened. And this, I think, is an important point where the construction was completed, and because we're working with government money, the plumbers were paid on fiscal year 2005 money. They charged all the pipes to demonstrate that there were no leaks, and then the building sat empty until everyone else did their piece of the work. I see Mark. Yeah, you know my world. And I think there were some issues. I mean, there are two issues that are clearly missing in this construction plan. Okay. So we think it's going to be in the pipes and the water at this point. We're getting it out of the clean water. I just kind of want to talk about the genomics part here. So we actually, our chief plumber at the hospital is an author on this paper because of all of this work that we did. We basically took apart sinks in the patient's rooms and went behind the wall to try to find where this was. And we can grow Sphingomonas carinensis straight out of the water and out of parts of the sinks, like these aerators. Okay, but how are we going to really track down the source? Because a lot of what people do is just pick one isolate, analyze it, and sequence it. And that's what we have from the patients. And I think that's appropriate where it's an acute infection. But in this case, where we're trying to find a point source, we actually can't miss the genetic diversity that is coming from there. So what we do, we really are very satisfied with these plate swipes, where you basically aren't just taking that one isolate. You take everything that grows on that plate and you sequence it with shotgun metagenomics. And then we also do sequence isolates which help us to phase the SNPs. So let's say on this plate I had, these four or five different strains, and I sequence each of them so that I can find the SNPs, and then I identify which are shared SNPs, and those could be useful for phylogenetic history, but for source tracking, I delete those SNPs, and I basically am just looking at SNPs that are specific to the isolates. So now if I get reads from the plate, I have all these different reads, but then I align them to the genome, and I'm basically seeing that I see, these blue SNPs and I see the orange SNPs, and know that that means that that plate had, 80% blue and 20% orange, and I'm kind of using that. So we're going to use that then to track back through the hospital plumbing system. So here's an example where out of the water, I'm getting 3 isolates. We then, as you saw, we're going to remove the sink from that patient's room, and the patient's room then goes negative for months. But 7 months later, the sink starts to go positive again. So we culture again, and we sequence everything on the plate, and we can identify the green and the blue strains based on SNPs. And the next month, we can see that the green isolate is there, the blue, is coming there at a minor population, but the red isolate is gone. So the red would be, colonizing the sink, but this proves to us that it's actually behind the wall that we have to get to, because, you know, the sink comes back recolonized with an isolate that was there before. So that would be our model. We also use this to find when we could identify the same strain from 2 different rooms, and postulated that the pipe that is feeding both of those mice then, be shared between those 2 rooms. So I think the SNPs that we find in these isolates are incredibly powerful, and they really, at this point, I would say that the genomic investigation really moves us beyond even just identifying what is this bacteria. I won't go through this. I mean, we also have a whole remediation strategy. We now no longer, we haven't had a patient for 2 to 3 years with Sphingomonas carinensis since we've remediated the situation.
Is that 140 Fahrenheit for 20 minutes?
Yeah. That's like what you do for Legionnaires. I mean, that's superheating the water and putting it through the pipes. Yeah. We don't actually use those chafing dishes. So anyway, in conclusion, I was just trying to give you stories of, how we track down emerging pathogens, how we do that in the context of our work on the skin microbiome, what we're seeing with eukaryotic viruses, and probably more of a classic indolent outbreak, and how we're using genomics to track back the point source there. So I tried to give people credit, but that's the great group that does this work with me. So thanks.